All right, hi guys. Another video. Gonna start Unit 5. This video. It's a short unit. There's only 2 lectures. The first of which is what this video is on. It's going to wrap us up on the nervous system. The lecture is about sensations. So something that we've been introduced a little bit into so far. So let's just dive in on it. All right. First definition is just what a sensation is. It's actually not the first time we've seen this. If you think back to the first lecture of the nervous tissue, the first lecture of unit number 4, um, we talked about the 3 functions or general functions of the nervous system, and the sensory function was one of those. So, nice little review already here for us. So, reading through. Sensation is the conscious or subconscious awareness of changes in the external or internal environment. We talk about external environment, we're talking about things outside of us, like, if we have an itch, if we're hot, if we're cold, if someone touches us. Those are things that are coming from outside of us, and we can be consciously aware of those. Changes internally to us could be like a change in blood pressure, change in oxygen levels in our blood, change of, pH. Typically, we don't have conscious awareness of those things, and that's because they're processed in a different area of the brain than the things that we have conscious awareness of. Having conscious awareness of sensations is called perception. That definition there is the second bullet there. Conscious awareness and interpretation of sensations primarily is a function of our cerebral cortex. Thinking back to our lecture on the brain, we learned about things like the primary visual area, primary olfactory area. That's what we're talking about here. As I said, we have conscious awareness of a lot of things. We don't have conscious awareness of everything. Regardless, the nature of the sensation and the type of reaction generated vary according to the ultimate destination of nerve impulses that convey that sensory information to our central nervous system. And I've already started to, but there are a lot of different sensations. I've listed off several of them: touch, pain, vision, hearing. Each of those is called a sensory modality, and we can group them into 2 major categories or classes: the generals and the specials. The general senses include both somatic and visceral, so there's 2 types of generals. Somatics include tactile sensations of touch, pressure, vibration, itch, and tickle. Thermals that include warm and cold, pain, and proprioceptive. We'll talk about each of those a little bit more here on the next page or 2. Viscerals we won't really get into in this lecture, but those provide us with information about our internal organs like pressure, chemical concentrations, stretch, nausea, hunger, and temperature. Hang on 1 second. Hello? Huh? Down in the basement? Yes. Okay. Well, I just started a video, so if you need me, I can pause it and start it over again or whatever. Bye. All right, these special senses, what we'll spend most of our lecture talking about, include smell, taste, vision, hearing, and balance. So we'll be talking about like the eyes and the ears and the tongue and that kind of stuff. Process of sensation, whether it's a special sense, a somatic sense, a visceral sense, it all begins with the sensory receptor. These can either be specialized cells, or typically what we see a lot of times is just the dendrites of a neuron. A neuron, in this case, we're showing a unipolar neuron with the body off to the side, is what's being directly stimulated, in this case, by a cold stimulus. So like, when you walk outside on a cold morning. And sometimes those dendrites are covered up with what's called a capsule. So we call them encapsulated nerve endings versus what we see up here as free nerve endings. It's the same thing, just this capsule, which can be like connective tissue, just makes it a little bit more sensitive, so it's easier to stimulate the dendrites inside. In this case, pressure would be what's stimulating them. And associated with some of our special senses, instead of stimulating the dendrites directly, what we stimulate is a epithelial cell called a receptor cell, or a separate cell that then synapses with the dendrites of that first order neuron that then propagates that signal to the central nervous system. Regardless, whether it's this type of receptor or it's these, the same result still occurs with the action potentials or nerve impulses being propagated into the central nervous system. So overall, the general purpose of a sensor receptor is just to convert the energy of a stimulus into the energy of a nerve signal so that the central nervous system can detect and interpret stimuli acting on and within the body. All right, let's talk about some of those somatics. Okay, somatic sensations arise from stimulating sensor receptors in the skin or the subcutaneous layer, the mucous membranes of the mouth, vagina, and anus, muscles, tendons, and joints, as well as our inner ear. Those that arise from our skin are called cutaneous sensations, and that's where we have a lot of them, which makes sense because our skin is what's exposed to that external environment. And so, we can interpret signals coming from the external environment, like hot and cold, stuff like that. The number of receptors for somatic sensations are distributed unevenly, meaning that, areas of, for example, our skin don't have the exact same amount throughout throughout the entirety of our skin. Places that are the highest as far as density goes would be the lips, the tip of our tongue, and our fingertips, which makes sense because those are the things that we are using to, touch and feel and experience the outside world. As we talked about on the previous page, there's 4 modalities: tactile, thermal, pain, and proprioceptive. And so I've listed off each of those and given a little bit of information about them. Tactiles include touch, pressure, vibration, itch, and tickle. Several types of what we talked about on the previous page: encapsulated mechanoreceptors attached to large diameter myelinated A fibers mediate the sensations of touch, pressure, vibration. Let me kind of walk through that sentence a little bit more. So we just talked about encapsulated means that there's a capsule around the receptors, around the dendrites. And this phrase here, large diameter myelinated A fibers, just refers to the size of the axon. Remember, the axon is a long wire of a neuron. The bigger the diameter, the faster that it sends a signal. We've learned about myelin before. Myelin, as we know, helps increase nerve signaling speed. And then we actually can classify them as A, B, or C. The axons, it just relates to their size. A's are the biggest, B's are the second largest, and then C's are the smallest. And other tactile sensations like itch and tickle are detected by free nerve endings attached to small diameter unmyelinated C fibers. So in this case, these would be sending signals a lot slower than these large diameter myelinated A fibers. Receptors involved in touch include corpuscles of touch, or Meissner corpuscles, as you'll see them. We find those in the dermal papillae of hairless skin. Remember, dermal papillae, if we think back to when we talked about skin, are the little outward projections of the dermis that kind of helped interlock with the finger-like extensions down from the epidermis to kind of give that zigzag jigsaw puzzle piece arrangement to our dermis and epidermis. Meissner corpuscles or corpuscle touch are only associated with hairless skin. So like, in our fingertips, which, the inside of our fingers, we don't have hair. Each corpuscle is an egg-shaped mass of dendrites enclosed by a capsule of connective tissue. Other places besides the fingertips include the hands, the eyelids, tip of the tongue, lips, nipples, soles, clitoris, and tip of the penis. All places that are extremely sensitive. Hair root plexuses are things we talked about previously. Obviously only found in hairy skin, and those are what wrap around the hair follicle down in our dermis and make our hair on top of our head or anywhere else that we have it basically an extension. And so that's why we can feel and sense when someone is touching our hair or when we touch our own hair. Merkel or tactile discs, also known as type 1 cutaneous mechanoreceptors, or touch receptors in the deepest layers of the epidermis, plentiful in our fingertips, hands, lips, and external genitalia. We learned about those when we talked about the skin back at the beginning of the semester. And the last type are type 2 cutaneous mechanoreceptors, or affinity corpuscles, down in the deep dermis, as well as ligaments and tendons present in our hands and abundant on the soles. A lot of those actually we can see here. In fact, we can see them all here in this illustration. On the left side of the page, we can see, hair plexus wrapped around the hair follicle. Type 2s that I just mentioned, corpuscles of touch I talked about, found in little extensions of the dermis projecting out on the epidermis. The type 1s I pointed out. Let's talk about thermal sensations. Thermals involve thermoreceptors. These are free nerve endings, which we can see some of those right here at the top of our picture. Two distinct thermal sensations exist: coldness and warmth, detected by different receptors. Cold, which are found in the stratum basale, the epidermis, respond to coldness. Warm receptors, which are located in the dermis and respond to warmth. Now, think about this. When we talk about hot and cold, there's only a certain temperature range in which we can stimulate hot or stimulate cold, because if we stimulate something super hot, we're not going to feel hot, we're going to feel pain. Okay, same thing if we feel something super cold. Speaking of pain, the free nerve endings associated with these are called nociceptors. And I always tell students, remember, no for pain, because most people will say, do you want to stimulate these? Their answer will be no. So it's an easy way to remember them. We find them in almost every tissue of the body except for the brain. And they respond to certain different things: intense thermal, so hot, cold, mechanical or chemical stimuli causing the release of chemicals, ultimately stimulating them. And then proprioceptive are shown over here on the right. These are what allow us to know where our head and limbs are located, how they're moving, and if we're not looking at them, so we can walk, type, or dress without using our eyes. Kinesthesia is our perception of body movements, and the receptors involved are called proprioceptors. There's three major types. Muscle spindles, these are the ones that are found within the muscle. They wrap around the muscle fibers and monitor the changes in length of a skeletal muscle. These are what are activated, like when you tap on the patellar tendon and activate that knee-jerk reflex. Tendon organs, or sometimes people in places will list them as Golgi tendon organs, found at the junction of a tendon muscle and monitor muscle tension. And then I don't have a picture to show them, but joint kinesthetic organs, or receptors, are found around the articular capsules of our synovial joints. Remember, articular capsules are the kind of connective tissues that hold together our synovial joint, and they monitor position of the joint and movement of that joint. Okay, so really, like I said, I'm not going to spend a lot of time talking about the general sensations. I'm going to spend the majority of our time talking about the specialists. So we're going to go through them individually, 1 through 5, starting with smell. And we're all very aware that our nose is what's responsible for the sense of smell. Within our nose, there's 10 to 100 million receptors for this chemical sensation. Fancy term for smell is olfaction. And those receptors are found within an area of our nose called the olfactory epithelium. It's in the most superior portion of our nasal cavity, covering the inferior surface of the cribriform plate, which is the roof of our nasal cavity, and extends along that supranasal concha, which is that one fold of it's a fold of bone actually, from the ethmoid bone at the kind of almost roof of our nasal cavity. In our olfactory epithelium, there are 3 major types of cells: olfactory receptors, supporting cells, and basal cells. The receptor cells are the most important for the sense of smell. The other 2 play a role as well. And the reason that these are so important is because they are the actual first order neurons, which are the ones that carry the signal from our nose up into our central nervous system. They are a bipolar neuron, which means the cell bodies in between the 2 extensions. And if you think back to our lecture on the nervous tissue, we learned that bipolar neurons were rather rare. The most important, kind of side for the senses, let me rephrase it. The whole thing's important. But as far as, the first generation of an impulse, let's talk about the dendrite. It's an exposed knob-shaped dendrite that is down in the olfactory epithelium. And then the other side, on the other end of the neuron, the unmyelinated axons project upward from the cell body and actually project through holes in that cribriform plate. These are called olfactory foramina, where they then can synapse with receptors or neurons inside of our olfactory bulb. The olfactory bulb being the fattened end of our first cranial nerve number 1, the olfactory cranial nerve. So when the picture is zoomed in, you can see those little extensions. Those are the those are the first order neurons, those olfactory receptor cells. There's the bulb. The signal is going to go through the cribriform plate and synapse inside, which is what we're showing off over here. Okay. Now, let's read through some more. There are 40 or so bundles of axons collectively forming the right and left olfactory cranial nerves number 1. These terminate in the brain in paired masses of gray matter called the olfactory bulbs, located just below the frontal lobes of our cerebrum. Oh, hang on one second. Sorry, guys. What's up? Hi, Bubby.
I didn't do this on.
What did you take your shirt off for?
You did it. Because I don't know.
Did your goofball head?
Mm-hmm. I was going to stay.
Did you sleep good?
I sleep in your bed.
I know.
Then I sleep in my bed. With 3.
You go back upstairs?
Yeah.
All right, love you.
Love you.
All right, sorry about that. Teaching from home right now is a challenge with a 4-year-old and a 1-year-old. But anyway, okay, so picking up the part of the olfactory receptors that respond to the inhaled chemicals are called olfactory cilia, which is another way to just call them olfactory hairs. And they are going to extend, and they're very tiny little extensions, look like little hairs, and that's why we call them olfactory cilia. Cilium is singular. They extend from the kind of knob-like dendrite of that olfactory receptor cell. The olfactory receptor cell is what's shown here in blue, and those are the little hairs extending from that. They project, their projections located in the olfactory receptor dendrites, touching the olfactory epithelium, lining the upper surface of our nasal cavity, and they are where the receptors that are stimulated by odorants are located. Odorants just being the chemicals that have an odor and can stimulate the olfactory hairs. The kind of tannish shaded colored cells are the supporting cells, and those do exactly what it sounds like. They're columnar epithelial cells, provides physical support, nourishment, and electrical insulation for the olfactory receptor cells. They also detoxify chemicals that they come in contact, or that come in contact with the olfactory epithelium. And then in these kind of purplish, darkish blue shaded cells are basal or basal stem cells located between the bases of supporting cells, and they produce new olfactory receptor cells by way of cell division. And that's because the olfactory receptor cells have a fairly short lifespan, only about a month before needing replaced. Also associated with the olfactory epithelium are the tubes that carry the secretions from our olfactory glands. Also known as Bowman's glands, produce mucus, which, you know, we all know our nose is loaded with mucus, but mucus in our nose is actually super important for the sense of smell because it helps with dissolving those odorant chemicals, which are these little dots in our illustration, and that helps them come into contact with those receptor cells. So they're very important for the sense of smell, not just you know, for giving us runny noses when we are sick. Okay, sense of smell, pretty simple. Taste is very similar, also known as gestation. It's also a chemical sensation. In fact, there's a lot of relationships that exist between sense of smell and taste, and we use both whenever we like ingest things. And that explains why if you get a cold and you know you're all plugged up, your sense of smell is not very good. Your sense of taste isn't that good either. Things don't always taste the same. There's 5 primary tastes that can be distinguished: sour, sweet, bitter, salty, and umami, which is a Japanese word, if I remember correctly, meaning meaty or savory. Any other flavor that you would ingest, because those aren't the only ones, would just be a combination of those 5. We're all very aware that our tongue is where our taste buds are, and the receptors for taste are found within the taste buds. Taste buds are also found in places like our soft palate, which is on the roof of our mouth, the pharynx, which is our throat, and then the epiglottis, which is the structure that protects us from choking. Each individual taste bud is an oval body consisting of 3 kinds of epithelial cells, and it's pretty much the same situation that we saw in our nose. There's supporting cells, there's receptor cells, and there's basal cells. The receptor cells, called gustatory or taste receptor cells, are, and I'll zoom in at our picture of a taste bud, kind of have a banana shape to them. They're kind of these bluish shaded cells, and at the tip of them are little hairs, or microvilli as they're labeled here, which are the actual receptors for the sense of taste. Those extend off of the receptor cells through what's called a taste pore, an opening at the tip of each of those taste buds, which we can see are found in the crevices of what's called a papilla. And we'll talk about papilla here in just a second. Okay. Now, unlike the olfactory receptor cells, gustatory or taste receptor cells are not a neuron. They are receptor cells that then synapse with the first order neurons in the gustatory pathway. And if I go back to this illustration, it actually shows an example of one. So they're not a neuron, but they synapse with a neuron and ultimately end up with the same result, the carrying of nerve impulses into our central nervous system. Basal cells, stem cells that function to produce new supporting cells, and the supporting cells are the kind of almost clearish shaded colored cells. They provide support, as it obviously states, but they actually will develop into gustatory receptor cells. So it's kind of cool. These basal cells become these cells, and these turn around and become these cells. As I mentioned, we find our taste buds in little crevices inside of what we call papilla. Okay. Papilla is the singular form of papillae, I believe is how you pronounce that. Little elevations that are found on our tongue. And if you actually look in the mirror and stick your tongue out, you can kind of see them. It's little tiny bumps. Now there's 4 types. Not all of them have taste buds. The first type, filiform, have tiny spikes without taste buds, and instead contain tactile receptors, so they allow us to have, the touch sensation of, our food on our tongue and that kind of stuff. They help us with that texture aspect of, taste, which is not the same. But, when we put stuff in our tongue and that kind of stuff, we do have that, I don't want to say it, we do have that feeling of texture, and that doesn't influence our taste. If you've ever felt the tongue of a cat, it's very, very sandpapery, and that's because they've got a lot of these. They kind of are spiky and increase the friction between our tongue and food to help move that food to our oral cavity. We find them pretty much all over the entire surface of our tongue. Foliate papillae, not super important in adults because they degenerate in early childhood, which leaves us with the last 2 that play a role in taste. Fungiform, which the word fungiform begins with fungi, which mushrooms are, and that's where their name comes from. They have kind of a mushroom shape to them. They're found all over the entire surface of the tongue and contain about 5 taste buds each. And that leaves us with valate, which kind of form an inverted V at the rear of our tongue, also known as circumvallate, contain up to 100 to 300 taste buds each, and contain up to half of our taste buds total in our human tongue. So those are the receptors and structures associated with the sense of taste. I'm actually gonna have to stop the video here, guys, because I'm being summoned by my wife to go take care of our children. So I'll pick up with another video on the eye and begin vision here in a little bit. Thanks.